Biology and Environmental Studies at Grinnell College in 2016. She has also worked as a field botanist technician for the Bureau of Land Management. Let's welcome Samantha.
Thanks, Paul. Alrighty, so today I'm going to be talking about hybrid maize in the genomics era, specifically talking about this observation of single parent expression, the model of complementation, and the phenomenon of heterosis. So first off, why are we even talking about hybrid maize? Well, first off, there's this really neat phenomenon in which if you take an inbred line like B73 or Mo17, they do all right, but when you cross them to create an F1 hybrid, those F1s do far better than you would expect based on the inbred phenotype. And this phenomenon is called heterosis. And you can see that that is true for both plant height as well as ear traits, among others. Additionally, we have been noticing this phenomenon and utilizing it in our agricultural production to greatly increase maize yields for quite some time. In fact, since the 1930s, as seen in this graph, where we have the time on the x-axis and the average corn yields on the y. And so as you can see, once they started incorporating maximizing this heterosis in the breeding programs, there was this kind of exponential increase in maize yields along with some other advents. So we've known about this phenomenon for quite some time, but we don't really understand the genetic basis or mechanisms of it. And so there's been a lot of people working on this, and we still don't have a great model for it. But with the advent of genomics and other high throughput methods, we can start looking at other things that we could incorporate into these models. And one of those things was transcriptomics. And so some researchers thought, well, potentially these differences, this heterosis, might be differences due to differential expression in the hybrids versus their parents or between two parental lines. And so in looking at these expression profiles, they found this kind of extreme observation, which they dubbed single parent expression. And that's where you have expression of a particular gene found in one inbred parent and the hybrid offspring, but that gene is not expressed in the other inbred parent. So that's kind of a really extreme example, really extreme observation. And people wondered, well, what could be the mechanism behind this? And so it's been proposed that this is an example of the complementation model. But the complementation model is just the idea that during that inbreeding development, to develop those parental lines, those were undergoing very, very small population sizes. And so they were undergoing a lot of genetic drift. And because of that, slightly deleterious alleles could fix randomly within those individual inbred populations. And so you could imagine that those slightly deleterious alleles are either base changes or insertion deletions within either the gene sequence itself or in regulatory regions associated with those genes that could then alter the gene expression, potentially knocking it out. And that because these are slightly deleterious and likely recessive, when you bring those together in the hybrid, parent one, which does not have those slightly deleterious alleles, could mask the slightly deleterious alleles in parent two and thus complement. So this was one of the models proposed to explain this observation. And so we've kind of taken this work and we're working on an experiment to take this a little bit further. And so we've got three main aims. The first one is, given this observation, can we see this occurring in hybrids spanning a greater divergence time? Because previously all this work has been done in temperate modern maize lines. So we haven't looked necessarily at temperate parents versus tropical parents, or even further like teosinte to some of our inbred lines. Teosinte is wild maize for the people who don't work on maize here. And then given that we find this observation, can we explain these findings with the complementation model? And that will require some genomics tools that we have been recently developed and just weren't available previously. And then given all of this, can we relate this complementation and single parent expression to this phenomenon of heterosis using Phenomics? So here's kind of an overall workflow of our experiment. We have 13 inbred maize lines that we're using to create a full diallele cross to get our F1 hybrids. And then aim 2 is focusing on that first question. Aim AIM-1, excuse me, is focusing on that first question. AIM-2 is focusing on the second, and AIM-3 is focusing on the third. So let's kind of zoom into AIM-1 of how we could even identify these single parent expressions. So when I say a full diallele cross, what I mean is that every single combination is being made between these lines, as well as in both directions. That way we can account for maternal and paternal effects. And so in this graph, you can see each of these boxes represents one inbred line. The colors represents the heterotic group, or kind of their individual population. So blue is non-stiff stock, yellow is stiff stock, green is tropical, the pink is a popcorn, and the orange is a sweet corn. And then each line is representing a cross that we'll be making. You'll also notice that there are boxes around some of these lines and not others. And that is because those that are boxed are part of the nested association mapping population, which is a very famous mapping population that has been used to discover a lot of and do a lot of genetic and phenomic work in maize. And my lab, in conjunction with several others, are currently working on reference quality de novo assemblies for each of these lines, as well as the other lines in the NAM population. And so we'll have access to some really high quality reference genomes for this work. And then this is a pipeline for how to identify single parent expressed genes, coming from this recent paper by Baldolf et al. Essentially, you have data generation as your first step, just doing that RNA sequencing and generating those reads, and then processing the raw reads, basically removing duplicates and low quality reads. And then you have to classify whether or not the gene is expressed or not expressed. So you can, that's a bimodal distribution. And so what they use is this model where they're just looking at whether or not the average read count of that gene, and they're trying to see, is there an expression change based on the genotype while taking into account other effects from either lanes or barcodes, or and also the normalizations needed for specific genes and samples. So given that, then they use posterior probability modeling to see, okay, given this gene and this genotype, and they use developmental stages, we'll be looking at different tissues, is this value above a certain threshold? And if it is, we'll call it an active gene. If it's not expressed. And then given that, you can say any gene I is a single parent expressed gene if it's active in one parent, it's active in both hybrids, so in both directions, and it's not active in that second inbred line. And since this is a phenotypic symposium, I want to skip AIM-2 and kind of focus in on AIM-3, which is this idea of taking all of these various F1 hybrid crosses and looking at their phenotypes, and then trying to see if we can link that to this complementation model. So as you're probably all aware, doing GWAS and QTL studies requires a lot of samples. And so a lot of experiments have already been done on these NAM lines. So we want to leverage some already published results. So these are three papers and studies that look specifically at three really important kind of suites of traits for maize, particularly for heterosis. There was one on plant height, which included the populations for NAM, as well as an IBM population, which has the lines B73 and MO17 in it. And they looked at, across 10 environments, 4,892 real families, and they evaluated them for plant height, ear height, number of nodes, as well as days to anthesis, and looked for specific QTLs associated with that trait within those lines. There was also another paper published from similar labs using both, again, the NAM and the IBM populations in 9 environments using the same number of real families, but they were specifically looking at the upper leaf angle, leaf length, and leaf width, looking for QTLs associated with those traits. And then a third study was looking at kernel traits, specifically starch, oil, and protein content of these lines. And they used the NAM mapping population, as well as another diversity panel, the 282 ILAP population. And they look specifically in 7 environments with 4,699 reals for those maps. We want to leverage that data. And given that they've already found some QTLs, we want to ask, okay, now that we've identified our single parent expressed genes, do we see enrichment for these around these known QTLs that are involved with these heterotic phenotypes? Because if they are related to heterosis, we should expect to see more of them around these QTLs or within them. Another thing we can do is look specifically for selection signatures or shared ancestry for single parent expressed genes. And we specifically want to do this for non-stiff stock and stiff stock heterotic groups, because those historically have been selected to maximize heterosis. We would, if complementation and single parent expression are related to heterosis, we would, and that if you have more complementation, that would increase heterosis. Then we expect that all the blue lines, these non-stiff stock lines, should share variants that are not found in the stiff stocks and vice versa. So we expect to see, if you have this Venn diagram of variants that are only within blue, only within yellow, or shared between blue and yellow lines, that we would expect most of the SPEs to be in these separate areas of the Venn diagram and less in that shared region. And so with that, I would just like to acknowledge all of my wonderful lab mates and my major advisor, Dr. Matthew HUFFORD, in their support of working on and building this project. And I'll take your questions.
All right, we got one right here.
So what are the tissues you're looking in?
Yeah, so we want to, so specifically a lot of the other experiments have looked specifically at root tissues. So we want to expand that to root and a leaf tissue, as well as a reproductive tissue, likely pollen grains. And we should have RNA sequencing for 10 tissue types for the parents. It's just generating it for the F1 hybrids. So we're looking at what's going to be the most feasible, given that we want to maintain similar environments, while also growing up so many plants at once.
Thanks.
I have a question about going back to your original premise of mutations and being complemented. The other half of that is that they could be suppressed. So what will you do to look for active suppression in the complementation model as opposed to being mutated? So they could be suppressed in trans,
right,
as well as activated. So every time you look at expression, it either goes up or down.
Right.
And that could be due to mutation or suppression by another gene.
Right.
So I'm asking, I guess, what have you thought about that?
Yeah, that is a really good question. And it's really difficult to address that. So this is kind of starting the exploratory analysis of just seeing, can we find the single parent expression? And then given that we find it across these wider range of tissues, does the complementation model explain it? And so this is just looking at, we're just testing this one small part of it to see if this one small part holds up.
What's the alternative explanation?
Right. There are other explanations. There's also the overdominance hypothesis, which is just that hybrids do have heterosis simply because they're heterozygous at loci and there's heterozygote advantage. There's also the epistatic model, which is that just by being in a different background, genes that were slightly deleterious are now slightly beneficial, or there's something about being in specific backgrounds that leads to heterosis. And there's no one model can explain all the observations that we have seen for heterosis. And there's a lot of overlap. So any model that you make for heterosis has to include basically all three of them, which is why we're trying to just focus on a small portion to then try and explain this one small portion, because trying to explain all of it is not feasible in 3 years.
Awesome. Do we have any other questions? All right, let's thank Samantha for her time and all